So the inferences here are all about causality. And in your mouse experiments, the kinds of things that Rob does, you can manipulate the mouse. But these are observational studies of buildings or barns. And how do you dissect causality from these observations so that we can figure out whether there's a way to intervene that's going to have a positive and not a negative effect?
So work that I didn't show is at the Biology and Built Environment Center. We're doing highly controlled experiments to get at causality, for example, by using small boxes. And this is to understand what drives the ecology of the built environment microbiome, not necessarily the human microbiome and human health. But we're putting dust into these chambers and then manipulating with very high precision while holding all other parameters constant, daylighting, for example, and looking at how that impacts the viability and the composition of the microbes through time.
So,
so I was going to say, for the human study populations in our hospital environment, what we've actually started to do is put intervention studies in place in the hospital room. So we are trying to limit their interaction with bed rails, which are a predominant source, we believe, of healthcare-associated infections. And we're also doing controlled double-blind trials on particular formulations of bacterial communities, a probiotic, if you will, that we add to the patient prior to their surgical practice and post-surgical practice as a dietary supplement. And we're doing that in a double-blind trial mechanism in order to try and determine whether it actually has an influence upon the outcome of their stay in hospital. You can't manipulate or control a human, but you can do a large enough statistical trial to determine the efficacy of a particular treatment.
do you have to get informed consent? Just
yes.
a question.
Yes, of course. Yes. For all of those, you need an IRB and informed consent.
I think this is mostly a question for you, Dr. Knight, but I work in obesity and its interactions with inflammatory pathways, but I haven't had time to really dig into the microbial evidence that we have, which is apparently really compelling. But what I'd like to know is whether or not it's been ascertained what the true mechanism is that these other bacterial populations have on obesity, and whether or not it's got anything to do with like endotoxins.
Yeah, so the mechanistic work has mostly been conducted in animal models rather than humans, as you'd expect. It depends somewhat on which microbial community you're looking at. So there's one community that's triggered in the OB mouse mutant model, where what it seems to be is primarily an energy harvest effect, where they're better at getting energy out of complex polysaccharides. The TLR5 knockout community, where fascinatingly it seems to be a behavioral phenotype. So the altered community causes mice to eat more, that's why they become obese. You can cure them with antibiotics, or you can cure them by just putting in the amount of food in the cage that a normal mouse would eat. And then there are some individual bacteria like Christiansenella, which in work with Ruth Lay and Tim Spector, we identified as one of the bacteria linked to low BMI in the UK twins cohort. And so that taxon is able to at least partially reverse obesity in diet-induced obese mice, and that seems to be linked to inflammatory pathways. So as you would expect from a complex multifactorial disease like obesity, there are many different pathways that microbes can be exerting their influence through. Some of those are inflammatory, but some of them seem to be more linked to behavior or to energy metabolism.
Can I just do a quick follow-up on that?
Sure.
And the other component is, we've been investigating with Eugene Chang at University of Chicago, and a number of other labs around the country have, is the circadian rhythm disruption. Mm-hmm. And so feeding, again, animal models on a high-fat, high-sugar diet significantly alters the microbial community structure, which leads to certain metabolites passing into the bloodstream, which then seem to interact with circadian functions in the liver. Now, the exact mechanism by which the disruption of circadian mechanisms in the liver affects obesity generation is still poorly characterized, but the same mechanism occurs if you become circadianly disrupted in the brain through jet lag or sleep disruption, you see the same signatures, metabolic and immune regulatory signatures being manifest in the body with that same mechanism. So disrupting the circadian rhythm of bacteria in the gut seems to have a significant influence on circadian rhythms in the body, dysregulating the whole system, leading to a disrupted phenotype, right?
Yeah.
So I had two specific questions for two of the speakers. So in terms of the forensic study, if the two burglars spent the same amount of time in each room, there really wouldn't be any way to tell if there was one burglar or two, right? It was only because you knew the signatures of the people beforehand.
Yeah, so and this is the flaw, right? And J. Edgar Hoover had the same flaw when he's tried to create a national database for fingerprints. In order to identify that it was a person in that room, you had to identify their fingerprints in that room. And we need a microbial fingerprint database, which I'm not advocating for, but would be necessary if we wanted to explore the applicability of that. But we could not identify the two individuals in that room until we have a signature of each of those individuals. That's
But
correct.
even if you had them, if the two things were combined, wouldn't it be hard to tell if that was a third person with a microbiome that was a combination of the two?
It would be very possible.
I don't know. I think the distributions. So this
You could do
characteristic
clustering.
abundance distribution, and no one's really systematically tested whether you can pick a combination of microbiomes out from a single microbiome.
Yeah.
It's plausible that you could do it, but I don't think anyone's done that specific analysis yet.
The American gut's helping with this significantly because it's enabling us to see the broad range and diversity of people's microbiota across thousands of people, and that will help with dissemination. But if I have a 116S ribosomal RNA or one metagenomic signature from one sample, disassociating it into individual signatures can only really be done if you're understanding the probability of particular strains being co-associated with a particular individual or multiple individuals. So yes, it's possible, but no one's really looked into it, as Rob said.
Yeah, and one issue with using the American Gut Database for forensics is that although we can now use UCSD's 501c3 status to accept donations to sequence underrepresented groups, at the moment, because it is participant supported, most of our participants are relatively well off. So I guess from a crime point of view, we're overrepresented in white collar crime and underrepresented in burglaries and so forth.
That's true.
I would add, though, that if the database is large enough, then even if you couldn't identify a particular person, you may be able to identify traits of an individual.
Oh, microbial profiling. I don't know if I like this.
And our National Institutes of Justice grant is exactly about that. And in fact, in that study, we were able to determine that even without looking at the burglar's microbiome, having that outside of there, that whoever invaded that property, one of them had a susceptibility to migraines, and it looked like they were actively taking migraine medicine. Because the migraine, and this is being followed up in a secondary study with Rob's group and my group, but the migraine medicine appears to have a significant impact upon the microbiome. So probably an effect on the microbiome rather than any causal relationship, but that leaves a signature pattern in the microbes we leave behind. And so there was a significant probability that one of those burglars took migraine medicine.
And
And
the
that's the number-
subway
that's the-
study that our data was part of also showed different signatures in subway stations that certain ethnic populations were in and everything else. So
That was the New York subway?
Yeah.
But that was in the human genetic element, right? The microbiome didn't leave any signature of that actual
it
trend.
was all the microbiome. It
There
was
was
a
a human
microbiome of the New York City subway.
But in that one, I thought that data came from the pit that left to the ethnic differentiation was from the human genetic material that's left behind in the metagenomes.
Oh, okay. I thought it was microbiome.
No, I don't think the microbiome had a signature of ethnicity in that respect.
Yeah, I think that was from the human reads.
Yeah, it was from the human reads, and the human reads part of that paper was very effective.
Okay. And then just quickly, I'm sorry, I'm taking a lot of time. All right, forget it. I'll ask you after.
Okay.
Okay.
Regarding these measurements of the microbiome and how they're affecting all these metrics of health, immunity, and even what we haven't really discussed directly today, there's a growing body of research talking about how the microbiome can influence, say, neurological disease. What are the molecules from these microbes that are interacting with the body? Are these molecules that are being secreted or shed, or these the dead microbes that are influencing these interactions?
Can I touch on that briefly?
Yeah, go for it.
Just briefly, for the neurological diseases, we're seeing relationships between bacteria in the gut produce GABA, they produce serotonin, they can produce chymeric acid. They can have a significant influence upon neurotransmitter levels in the body, in the gastrointestinal nervous system. And also some of the other molecules that are being produced can pass into the vagal nerve system because there's no blood-brain barrier, leading to significant alterations in neurological signatures between the gut and the brain. So there are that plus immune regulatory elements and its impact upon neurotransmitter pathways can have a significant causative influence.
There's also some work that's coming out very soon that demonstrates that even inoculation of certain dead bacteria can have substantial neurological effects in mice. And that hasn't been replicated in humans yet, but it's a very exciting direction.
And one last remark, Rob Knight, your coursera course on the gut microbiome, just finished it, and it was fantastic. Just wanted to say that.
Thank you.
Excellent presentations from the whole panel. But this question is from Erica, or for Erica. Did you look at antibiotic exposures in the different populations, and could that explain some of the differences in allergy in the farm children?
Yes, we looked at the antibiotics exposure of the children, and we did not see much of an effect. Now, I think the antibiotic, there's a lot of indication that antibiotics may work for obesity. However, and I think that's something that needs to be stressed here, a lot of this evidence from the microbiome comes from animal studies. There is still very little that comes from human studies. And I think we need to have a little bit of caution to extrapolate everything we see from mouse to humans. I still think there are differences. We know that the mouse microbiome is different than the human microbiome. And so I'm not so sure whether we can basically transfer everything. The antibiotic story in children is different because children get treated because of asthma with antibiotics, and therefore there's a positive association, and you cannot tell, is it because of the disease or is it really because of the antibiotics? I think we need to do much more studies, well-controlled studies in humans, and to look at large sample sizes, not just 20 or 30, and to really try to link the two. And then I think Jeff Drazen's comment on where's the causation, where's causality here is, I think, the most important. At the end, yes, we want to change the environment, and I'm heavily pro this, but what exactly, which are the microbes that are going to be beneficial? What is it, diversity? If we're talking diversity, it's a completely different set of ideas than just if we add a few probiotics. I mean, there's so much to be discovered here that I think we're at the beginning.
So one thing that I was just interested in was how quickly it seemed like the shape of or the types of bacteria changed over time, because like people leave a building and over like a day or two, it looks like the structure totally changes, and like 12 hours later you get the signatures of these people who are potentially burglars, but then maybe later it doesn't really last the same. So my question is, why does it change so quickly? Is it because like these houses are not like bacterial incubators? And then the other question is, like, are there potentially spaces that are much more like long lasting? So if you wanted like a good time scale that was longer than a day, could you like look underneath like a warm couch or something like that, and it would actually last like a week or something like that?
I mean, vast majority of bacteria which are released into this environment die pretty much immediately. I mean, there's very few of them which have the capability to survive. One of my ex-students just wrote a brilliant and rather scathing report on the microbial wasteland of the built environment, which it is unless it gets wet, unless it gets warm and wet, and then things can go very bad for fungal and bacterial population growth in terms of human health. But the instance of the burglar report, we can track microbial signatures in the building in which those burglaries happened, and this is research we haven't published yet, but it's new analyses, up to approximately 36 to maybe 44 hours. We can't quite get it any higher than that. We start to see it decaying rapidly after 2 days. And so that entirely is dependent upon the activity in that house. If if two people burgled your house and then a load of people moved in and had a party, the signatures would probably be completely eroded within hours. And the erosion is because pure deposition. 38 million cells being released into from each of us. The previous people up on this panel, their microbiome signature could have easily been eroded by Erica and me just waving our arms around, So we have to be very careful of that. As to where the microbes grow better, I don't know, Jessica?
Well, I was actually going to comment on something you said earlier. What we've found at the Biology and Built Environment Center is that it's not just occupancy that matters in terms of the temporal dynamics of what's going on, but how that environment is designed is going to influence how the bacterial community changes through time. Yeah. And what did you ask me something specific?
Oh, like
I was speaking
what grows
more
where?
to what grows
Yeah,
where.
and what's the best environment for maintaining a signature?
I don't think we know the answer to that question, and I actually don't think we know that viability is only coupled with moisture. I think that there are other environmental factors that may play a role.
So we have time for 1, 2, so make this, we'll start over there. Fast question.
Having way too much information about what's out there, how has what you know and what you look at changed your own behaviors from carrying around a shower of Purell or bringing an Amish cow into your house?
We rescued dogs. So my wife made me rescue dogs after we saw some of Erica's commentaries a while ago, and then our own work in our own home. We saw a paucity of animal-associated bacteria, and so we went and rescued a dog from Kentucky, and just recently we rescued another dog. And so we're trying to increase the animal quotient in our homes. Also, we're part of the Amish and Hutterite study, and my kids go and regularly visit the Amish farm in the summer now, just for like 2 or 3 days, and they do work on the farm. I'm teaching them a lesson,
But you keep them away from the Hutterite farm.
Yeah, the Hutterite farm.
I actually want to hear what Erica has to say in response to that question.
Well, I think it's an excellent question because we cannot turn time back to lifestyles that were lifestyles of my grandparents. I like the approach that Jessica takes because I think if you think of the microbiome, where does it come from? It comes from people, it comes from animals, it comes from plants, it comes from soils. And maybe it's not a good idea to just keep it outside of our living spaces. And so I think to think about, what have we done in our modern buildings, I think is a good idea without telling exactly what is possible. In my part of the world, what people do is that they have now kindergartens in farmers' places. And so that's maybe, in my part of the world, that's something that can work. I don't think it can work in Boston. But try to understand what is the need to, how should I phrase that? If I would put this in a larger frame, I think we need to understand each disease. And in my view, asthma and allergies is something that needs activation by innate immunity. And probably it needs a lot of activation constantly by signals that are sort of outside the body. And we have forgotten that there's things that are outside that somebody needs. It's like the microbiome in the gut is something that does our vitamins. And you would think vitamins are so essential to our health that nobody would outsource and accompany the production of vitamins, yet it's happening. And so what if in our outdoor environment there's something that is also outsourced that we need? And I think this needs to be understood. I think we're just at the beginning. I think all of this is fantastic and highly interesting. But what is that interplay? What is that interplay not only between microbes, but between our immune system? Is it that our immune system needs certain things, not just reacts because they are bad? This was the constant idea, but maybe I think what's changing is to say there, as Jessica pointed out, it's not just pollutants. Maybe there are some things that are good, and maybe some things that we need. And I think this is what we need to understand. In terms of what can we do immediately, well, I'm a researcher. I think we need to do research.
I get to ask a cow question. I'm from Bishop, California, which is a small town in the eastern Sierras. And we have some miraculously pristine streams that flow out of our mountains that have been extremely well studied by Los Angeles Water and Power, our SNARL, our Sierra Nevada Aquatic Research Center, and our city officials. And they are extremely concerned because by the time this pristine river reaches our city, all of a sudden it begins to spike in coliform counts. And they've actually recently now had the ability to test, and they're finding that most of these coliforms are coming from the cows that live around town. I used to have a cow that lived on the other side of the fence from me. And they're completely horrified about the health benefits because they're concerned, this violates regulations, blah, 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 blah. They're putting signs in all the rivers, don't swim because of high bacterial count. Is this information actionable enough that I can go home and tell them to lighten up? And also, as soon as the water leaves town, it gets very, very clean again.
Well, I think the new concept which the microbiome has introduced into research is about equilibrium. It's about, what's the equilibrium between germs that we need and germs that are bad. We have focused so much on the bad guys. And what is the ecology of all of this? It's not just about one germ, about one cow. Yes, if the cow is diseased, yes, you're in trouble. If the cow is maybe not diseased, maybe it's a good thing to have. And I think this is a new concept also in research and in science. How can we have a truly ecologic approach to this? We have been so used of identifying one bad thing, or it goes from A to B to C, but I think it's much, much more complex. And we're all, I think, starting with this and all this omics technology and all the biostatistical tools that we're having now to really have this big data, trying to understand this. It's much more challenging. And I think we are, and what we heard by Mike Snyder this morning is so interesting because you see, if something happened, you have a cold, you have, I don't know how many genes that go up. It's not about one thing. And we're not used in medicine on how to identify these networks. How are we going to do this? I think there's so many open questions here. And so, again, as you can tell, I'm a researcher.
Is your information openly accessed so I can take some home with me? Or
sorry?
Is your information published in open access so I could share it when I get back to my community?
Yes. You find everything on the internet. Yes.
Thank you.
Yes.